the, okay, let's go here. Oops, hang on, my arrows are not working again. Just give me a minute here. Oh, there we go. Okay, it's working. Okay. All right. So everyone can see. So thanks for giving me the opportunity to talk about some of this talk. I've tried to pitch towards some of the embryology research that I've done in the past. It's part of the work that I did when I was doing my postdoc at the University of Utrecht in the Netherlands. But it's very much on embryo research. And given the focus of this subject, I thought this would be a good talk to give to you guys. So when I was in Utrecht, I was based at the Faculty of Veterinary Medicine, and I was based in an equine sciences department. So we were really interested in horse reproduction, embryo development, and certainly we were interested in what happens when you produce embryos artificially, so by in vitro fertilization. And so we knew that some of these in vitro fertilized embryos were not very viable. So we actually decided we would actually try to understand what was going on there. So today's talk, we're going to focus really on the effect of in vitro production itself on the horse embryo. And just before I begin, I'll give you a little bit of background about who I am. So I'm a associate professor at James Cook University. I deliver most of the reproductive physiology components to the medical students, to the veterinary students, and to the biomedical students at the university. And in terms of my background, I'm actually a scientist by training. So I've got a Bachelor of Science degree from Monash University in genetics. And then I did a PhD at the University of Melbourne in reproductive biology and was specializing in wildlife reproduction. And then I did a couple of research positions at the University of Glasgow in Scotland, and as I said just now, at the University of Utrecht in the veterinary faculty there. So I've predominantly got a research background in terms of my training. And when I finished those positions, I got a job at James Cook University, and I set up my own research lab. And we basically called that lab the GAME lab, the Gamete and Embryology lab. And so as part of that lab and its research focused, I developed a couple of different research interests. One of those was really just sort of understanding something about the impact of all sorts of different adverse environmental conditions. So in vitro production, things like heat stress, social stress, disease, even maternal age, and what effect that has on both the sperm, the egg, and the embryo. And I've been working in a number of production animals, but also in wildlife species. And in addition to that, I guess my passion has really been developing assisted breeding tools for animal production, but mainly for wildlife conservation. And so I've worked with marsupials. I've been lucky enough to work with some endangered African wild dogs, corroboree frogs, and so on. And so we were able to produce offspring by artificial insemination. We've developed sperm freezing techniques and all sorts of things. So that's really kind of the background of myself and the group. And in terms of the sort of approaches that we use a lot of sort of cell biology tools, some reproductive tools, and molecular tools to basically tell us something about the health of these embryos that are being produced. Are they healthy? Are they unhealthy? What's causing the problems? And I guess in the lab, we've got a lot of expertise in different areas. Some of those are essentially in the ability to collect sperm and evaluate quality, the different types of sperm quality. And so we use computer-assisted sperm analysis for this. We use microscopy and fluorescent flow cytometry. And we can sort of quantify these different states of sperm quality, if they're damaged, if they're dead or alive, if they have normal morphology, and so on. And we also have tools to look at the quality of the oocyte, the female egg, and the embryos that are being produced. So we can do viability stains, we can look at morphology, we can even track individual heart rate of developing embryos. These are some fish embryos that we work with. and we can do some fluorescent protein staining work as well with those guys. And then I guess at the end of the day, we also work across a number of different species and have tools to actually facilitate pregnancies and the fertilization of embryos. So we can certainly collect semen from multiple species. We can freeze it. We can use artificial insemination to get pregnancies, and we can also generate embryos in vitro by IVF. And those are the tools that help us investigate various questions in reproduction. And most of, you'll tell from all of these pictures here, even though I'm in the School of Medicine, I actually do all of my research is really in animals, and you know, deliberately so, because that's what I find the most interesting. And so we've got different projects that we're working on at the moment. Some we've been working. One of my PhD students was from the Philippines. Anita might remember Santiago, perhaps. I'm not sure. He was at James Cook, and we looked at the impact of heat stress on sperm quality and DNA damage. Work I'm doing at the moment is with an endangered African wild dog species. We're trying to set up sperm freezing and artificial insemination to manage their genetics. And I've done a few projects working with aquaculture species, so commercially important species there. Endangered corroboree frog, looking at disease effects on fertility, and in Thailand, working with a couple of colleagues trying to develop a sexing tool for very young turtles, so we can sort of assist the turtle the turtle industry, I suppose you could say. So that's the kind of that's a little bit about my background. And apart from that, we'll move on to what the talk is today. So the way I've structured this talk, there's a little bit of tutorial, a little bit of background explanation about basic processes of embryo development. This might be familiar to you already, but then I'll sort of move on and focus on the research components that come from that. So really to start off with, we'll just talk about this period of pre-implantation embryo development before the embryos implant into the uterus. We'll look at the development of those embryos to the blastocyst stage, and then we'll actually take a side turn and we'll look at what happens in the horse in terms of in vitro embryo production. We'll look a little bit about the gene regulation of the development of the blastocyst in different mammals, and then that should set us up to understand the research, which is about understanding something about normal patterns of gene expression in horse embryos to tell us whether or not in vitro embryos are healthy or not compared to in vivo produced embryos. So I'll talk a little bit about that today as well. And so, I'm assuming that you have a certain level of knowledge. Anita told me you're about 2/3 of the way into your embryology course, so you've probably covered a lot of this already, which is good. This period of pre-implantation development is essentially all of the embryonic development stages that occur while the egg or the oocyte is traversing the oviduct before it implants. And so really after about 24 hours after fertilization here, the embryo starts dividing 2 cells to 4 cells to 8 cells. And then ultimately, as it makes its way down the oviduct, it forms a clump of cells called the morula. And then just either before or as it enters the uterus, it differentiates into this early blastocyst type structure. And then typically hatches out of this structure here called the zona pellucida, which was the barrier to sperm fertilization. It hatches out of that, and then this hatched blastocyst now has an outer layer of cells that are available to start that process of implantation into the uterus. Okay, and what I'm going to flag here is that the timing of all of this whole process is important, because essentially that blastocyst needs to implant or attach to the uterus and send signals back to the mother to tell the mother that she's essentially pregnant. And those signals, what they do is they act on the corpus luteum and they keep progesterone functioning. So timing is really important with these embryos so that they can stop the loss of progesterone and the end of pregnancy. So they've got to develop at a certain time. And so these signals that they produce are called maternal recognition of pregnancy signals. So I'll keep on talking about MRP signals in this lecture because it's sort of important. All right. So, and these cells, as you may know already, they go through those cell division stages. Cell division in the embryo is unique. The cells divide in size. They almost halve in size, and there's not really a growth phase. So they go from sort of 2 cells to 8 cells, 16 cells, and so on. All of these early cleavage stages, up until the 8th cell stage, are what we refer to as totipotent. What that means is that they can actually, if you separated each one of these and you transferred them into a recipient animal, they would each develop into a completely new individual, almost a clone of one another, including all of the placental tissue and everything else. So they can differentiate into everything. Then they get to about 16 or 32 cells in number, and they're referred to at this stage as a morula. And so we've got a sheep morula, a horse morula here, and this is the first time that the cells kind of have different positions in the embryo. So some of them are stuck on the inside and other cells are stuck on the outside of that embryo. And that position will eventually determine their future fate. Once they go a little further, they get to the 64 cell stage here, and they start to look like this. They actually undergo their first differentiation event. And so the cells end up forming a clump of cells on one pole here. That's the cow blastocyst. And there's another clump of cells here in the horse, the inner cell mass. And basically that population of cells has differentiated. It's still what we call pluripotent. So it will form all of the tissues of the developing embryo, except now the outer sort of trophectoderm or the trophoblast layer. And that's this layer of cells around the outside here. Same thing in the cow here. That layer on the outside there is going to form the trophectoderm or the trophoblast, and it's going to form the future placental tissue, the chorionic tissue. And of course, that outer layer, apart from forming the placenta, has this important role in producing those maternal recognition of pregnancy signals. So it's a pretty important layer for continuing pregnancy. All right. And so I just want to focus on this transition in more detail, because the research that comes is really all about the development of these 2 sort of particular stages. So here's the morula stage embryo again. You've got these balls of cells, some on the inside and some on the outside. And this is really the future differentiation pathway of this embryo. So all of the cells that are on the inside interconnect with these gap junctions here. So there's sort of communication junctions between all of the inner cells. The cells on the outside here, though, connect with tight junctions. They're sort of very robust membrane type connections that are sort of almost epithelial in a way. And so you get these differences in the connections. And at the same time, those outer cells start to pump sodium ions into the intracellular spaces of these morula. And that creates this sort of sodium gradient, which causes the embryo to actually absorb water. So water basically diffuses into the embryo and causes it to swell, starting the formation of this blastocyst. Okay. And sort of as that blastocyst starts to sort of expand, a little bit like a balloon, there are other genes that turn on that start the differentiation process. Some of them actually, they start a little bit earlier. So in those outer cells here, in the morula, they already start to express a gene called CDX2, so caudal type homeobox 2, CDX2 is enough. And that turns on in those outer cells, and it differentiates them into these trophoblast cells, the chorionic placental tissue that's going to form. So CDX2 drives that differentiation. The cells on the inside start to increase their expression of another gene called OCT4, octamer binding protein 4, and that differentiates those cells into the inner cell mass, which is the future embryo, and so on. And of course, this embryo keeps on growing, it keeps on expanding, and it puts pressure on the zona pellucida, and the trophoblast or the trophectoderm here will start to digest away some of that zona, and the embryo will hatch out of the zona pellucida and start that process of attachment. So that's kind of the early development stages before implantation. Okay, I'm going to change direction for a minute and draw your attention to the horse industry to start with, which is really where the focus of this research talk is about. And so I don't have figures for Indonesia, but certainly in Australia and the US, the equine industry is worth billions of dollars. It's a highly valuable industry. $6 billion in Australia, $112 billion in the US in terms of revenue generated from this industry. So it's a big industry. And in terms of in vitro embryo production, it's not applied in the thoroughbred industry, but it's certainly applied in the equestrian industry. So you can produce offspring artificially in the equestrian industry, and that's worth big money. So people will actually pay a lot of money for embryos that come from champion mares or champion stallions. Okay, and so to some degree, in vitro embryo production enables you to sort of maximize the production of offspring from certain types of very valuable horses, either they're performance champions or they're genetically highly valuable. And so, for example, you might be able to, if you have a champion mare, she's still competing. She has, a window of maybe 5 or 6 years to compete. And so if she falls pregnant and carries pregnancies to term, then she's not able to compete. And so often in vitro embryo production enables you to collect oocytes from these champion mares, fertilize them in vitro, and transfer them to recipient mares so that the recipient mares can carry the pregnancies to term while the champion keeps on competing. Similarly, you might have, again, valuable animals that are now sort of, reaching the end of their reproductive capability, or they have some sort of uterine disorder that prevents pregnancies from being carried to term. And so collecting eggs, fertilizing those and transferring those to recipients means that the genetics of these valuable animals can also be rescued. Similarly, if you have a stallion with poor quality sperm, there's no way that stallion will be able to fertilize by natural mating. So if you can collect the sperm from that male and fertilize eggs in vitro, you can still propagate that highly valuable animal. And the same sort of thing, you might be in a situation where you've got these really highly valuable animals, either performance animals or genetically very valuable animals, and then all of a sudden they've broken a leg or they've had a bout of colic and they can't be helped, and so they need to be euthanized. And so in that way, in vitro embryo production enables you to collect the sperm, salvage the ovaries and the oocytes, and still produce offspring from these animals, even though they've just passed away. So it has quite a lot of value in improving the genetic quality of horses. And so there's essentially two methods for in vitro embryo production, sort of a conventional in vitro fertilization approach and a sort of intracytoplasmic sperm injection, so a microinjection. And I'm assuming you've talked a little bit about these already. Is that correct, Anita? Yeah.
Yeah.
Yeah, okay, so a little bit. Yep, okay, good. So I'll just talk about both just to give you a bit of a background. But so conventional in vitro fertilization, relatively straightforward. This is a human uterus, apologies there, but not a horse uterus. But you can recover oocytes from follicles from the ovary, culture those in vitro, mature them in vitro, introduce sperm, enable that fertilization to occur in the petri dish. So really the sperm are able to penetrate and fertilize the oocytes naturally, even though the environment itself is not natural. And those embryos you can culture in that petri dish. Typically you culture them until the blastocyst stage, and then you can transfer those into the uterus where they're ready to implant. So you transfer them back to a site where they should naturally be found at that stage of development so that they can then start that implantation process. So that's the standard sort of conventional IVF approach. The problem with that sort of straightforward IVF approach in horses is that it's essentially useless. Many studies have been conducted that have tried to produce offspring by conventional IVF, and it's been unsuccessful. And some of the reasons given are that capacitation of sperm in vitro is a problem, and also the zona pellucida layer that surrounds the oocyte, apparently in culture, in horse oocytes at least, tends to harden. So it makes it more difficult for the sperm to penetrate. So natural fertilization is quite difficult. in vitro for horses, at least. And so essentially the industry has focused on producing embryos by intracytoplasmic sperm injection. So in that case, it's basically hand-picking a sperm. It doesn't have to be incompetent. It can be a normal functional sperm, hand-picking that sperm and then using a pipette, directly injecting that and puncturing the oocyte itself, injecting the sperm into the cytoplasm of the oocyte. Now that's fine, but because that whole natural process of fertilization has been bypassed, the oocyte doesn't even know it's been fertilized. So it needs to be chemically activated to start that normal development process. And so after you do that, then the embryos will develop normally. So that's the alternative approach, which is really used in the horse. And so let me just go forward. Yep. Okay. Now, so in vitro embryo production is used in the horse, and a number of studies have compared in vivo produced, so naturally conceived, versus in vitro produced embryos. And some of those studies have found that there are problems with these in vitro produced embryos. Certainly, they're smaller than normal. They have less cells, and they have high rates of chromosome duplications and some cell death as well. And just to sort of show you the numbers here, this table's a nice one. It just shows the naturally conceived embryos here when they looked at 10 of these embryos and they compared them to two different groups of in vitro produced embryos, compact cumulus oocytes and expanded cumulus oocytes. Don't worry too much about that at the moment. When they compared the in vivo to the in vitro produced, what they found on day 7 in both of these embryos is that the naturally produced embryos were almost twice as big than the in vitro produced. So they were larger. They had much more cells than the in vitro produced embryos. And most of the cells in those naturally conceived embryos were just purely diploid, whereas only 60% of the cells in those embryos from the in vitro produced source were actually diploid. So they either had triploides or quadruploides or whatever. Also, the amount of damage that was found in the cells, even though these are low figures, was significantly higher than in the in vivo produced embryos. So in vitro embryos are not perfect, and it looks like they're developmentally delayed. And to sort of show you this a little bit further, here's there's a day 7 in vivo produced embryo, so a naturally conceived embryo versus a day 7 in vivo produced embryo. And the first thing that you notice is that day 7 in vitro produced embryo is developmentally delayed. It looks more like the day 6 embryo that's produced naturally. So it's already a day behind in its developmental potential. The other thing is, if you look, this is stained with an actin cytoskeleton fluorescent marker. And basically in the in vivo produced embryos, that cytoskeleton is quite distinct. If you look in the in vitro produced embryos, the cytoskeleton is more fragmented. It's less distinct. So the actin cytofilaments are not so great. There's another feature in naturally conceived embryos. They produce this green glycoprotein capsule here. And that's important for horse embryos because their implantation is delayed. And so normally they spend a lot of time in the uterus moving around. About 14 times a day, they move and circulate throughout the horse uterus. And they do that so that they can secrete this maternal recognition of pregnancy signal to maintain the pregnancy. So being mobile is really important. And having this glycoprotein layer is kind of a protective layer that keeps the embryo intact. Now, if you look at a day 7 in vitro produced embryo, there's two examples here. You can see that green fluorescent glycoprotein capsule is struggling to form. It's not really even coalescing around the embryo, and this is a day later than it should already start to appear. So the horse embryos here are those in vitro produced embryos. They're just not normal. They are delayed, it looks like. Now, the interesting thing is, if you transfer these in vitro produced embryos, you've got a pregnancy rate of about 70%. So that's actually quite high. The problem is, after the transfer, the embryos don't survive. And so there's about 50% of in vitro produced embryos are lost after transfer, compared to about 20% of naturally conceived embryos. So there is a problem with these embryos. And certainly, from what I've sort of shown you here, a lot of the issues that we're seeing are really in this developmental stage, going from the morula to the blastocyst stage. They're just delayed. It doesn't seem to be progressing in a timely way. And so just to sort of think about that process of differentiation from the morula to the blastocyst and the genes that are involved. I'm not sure if have you spoken a little bit about the gene regulation of early development, Anita, at all?
Not yet, actually.
Not yet. Okay, so this stuff, there's lots of genes, but these are some of the core ones. I'm just going to keep it simple and focus on these. But there's been some early work that was done, in the early 2000s, that basically determined that there were at least 4 genes involved in some of these early differentiation events. So going from my little pointer here, going from these morula stage embryos here through to the early blastocyst, and then to the expanded blastocyst that has an epiblast and a hyperblast layer. There are genes that drive that differentiation process. And so a lot of knockout mouse studies have really shown that these are the genes that are involved. And so certainly in the morula here, those inner cells that produce OCT4, those will give rise to the formation of the inner cell mass here. The ones on the outside that produce CDX2, those will go on and form that outer trophoblast. So that's these 2 stages here. Thereafter, there's sort of a mix of different genes turned on in different cells in the inner cell mass. NANOG in red will drive the differentiation of this epiblast layer, which will then go on and form the proper embryo. Whereas another gene here called GATA6, shown in green, those cells, when they turn those genes on, they'll go on and form this hyperblast layer or primitive endoderm, which forms the early yolk sac. Okay, so there's at least 4 genes involved in the mouse that drive this process. However, other studies in other species subsequent to this have demonstrated that some genes are preserved, but other genes are different. And so certainly in the cow, if you look at this process of development in the cow, OCT4 is expressed in the inner cell mass, but it's also expressed in the outer trophectoderm as well. So we don't think OCT4 drives that first differentiation event in the cow. And if we look in the pig, same sort of thing. So OCT4 is expressed in the inner cell mass, also in the trophoblast, but so it doesn't seem to have a specific role for the inner cell mass. And then subsequent to that, where we would expect NANOG to be expressed to drive this differentiation of the epiblast, NANOG doesn't even come on. It's not even expressed at this time. So it suggests that at least in cows and pigs, there's something that they do that's different. And just to sort of show you this in a more visual way, there seems to be these species specific differences in the genes that control the formation of the blastocyst. And so this is a mouse embryo, and it's immunofluorescently tagged for OCT4. And so all of the cells in the inner cell mass here are strongly expressing OCT4, and that's exactly where it should be. Okay. When you look at the cow, this is a cow blastocyst, and it's been rotated on its side. And so here's the inner cell mass here. And then, so they're all fluorescing OCT4 as well, which is where it should be. But then you've got all of these, the nuclei of all of these trophoblast cells also strongly expressing OCT4. And they shouldn't be doing that, because OCT4 is supposed to be a pluripotent gene for the inner cell mass. This should be trophoblast, so it should be just expressing CDX2. Same thing for the pig, inner cell mass, nice and fluorescent and nice and yellow. Then you've got these trophoblast cells on the outside that are also showing up green, also showing up yellow. So OCT4 is also being expressed in those layers where it shouldn't really be. So there's basically species-specific differences in the genes that control these early differentiation processes. And so that sort of made us ask a question for the horse. Since we knew that a lot of the developmental problems in vitro produced horse embryos were really occurring in this morula to blastocyst type of developmental transition, and we know that these genes are involved in that process, we thought, well, maybe if we looked at gene expression in these embryos, maybe abnormal expression of the genes might explain why in vitro produced horse embryos are not normal. And so we kind of set out to answer that question. But firstly, we didn't even know what was normal for horses. So we thought, let's start by looking at the normal pattern of gene expression in horses. And then we'll compare the normal pattern with these in vitro produced embryos to see if they're still normal or they're abnormal. And so this is where we come to the veterinary side of things. Anita, is your cohort, are they predominantly vets or medical students or biomedical students?
No, they all just biology student.
Biology,
Just
okay.
basic, yeah.
All right. Yeah, good to know. So this is the part where me not being the vet, I wasn't doing the animal work, but I was working with the vets to actually collect these embryos, which was really cool because you're standing in a stall with a horse that's like 6 feet tall, and you're collecting embryos from these animals, which was pretty amazing. And so essentially, this figure here shows you the timeline of embryo development in the horse. So ovulation on day 0, and they spend 6 days that embryo developing to a morular stage embryo as it travels down the oviduct. And by day 6, they reach the uterus for the first time, and they look like this. So these are those clumps of morulars, these early stage embryos. And that's really the first time you can collect these embryos by non-surgically flushing the uterus. It's called uterine lavage. And so that's really a good time to collect these embryos. So day 6 is the earliest to get morular, and we got 5 of those. We flushed a little bit later on day 6.5, and we got these early blastocysts, about 7. This took a lot of work, but anyway, we got these numbers. And then, we flushed again on about day 7.5, and we managed to get these later stages, these expanded blastocysts. Okay, so we collected those all up and we snap froze them. These were naturally produced embryos. So we froze those so we could then look at the gene expression. And of course, we wanted to compare those to in vitro produced embryos. And so all of these embryos here have been produced by ICSI, that microinjection. Okay, and so because we know that they're developmentally delayed, we had to sort of morphologically make sure that we collected an embryo that was a morular stage embryo that was produced by IVF, looks exactly the same as an in vivo one. So we're sort of comparing the same things. So we did that for the morular, we did that for those early blastocysts, and we did that for these expanded blastocysts. But in IVF, they look like this. So here's your blastocyst, and it's actually starting to hatch out of the zona pellucida. If you physically remove the zona, it will expand and it will actually look almost identical to the in vivo expanded blastocyst. So we collected the same embryos in vitro and in vivo so that they were the same thing we were comparing. All right, so we collected them all. Oh, hang on, I'll go back. And we snap froze them because we wanted to look at gene expression. And that's usually done by looking at a messenger RNA, which is a very unstable molecule. So we collected it and we had to quickly convert it into cDNA, which is a copy DNA. It's a more stable type of DNA. And it's basically a reflection of the genes that are being expressed at that particular time. And then we tried to look at there's a whole pathway of different genes you can look at in terms of embryo development, but we chose a few, and I'm only going to talk today about a couple. OCT4, that one that codes for the inner cell mass, NANOG, the one that codes for the epiblast, CDX2 that forms that outer trophectoderm, and GATA6 that forms the primitive endoderm. So we pretty much focused on those 4 genes, and we also had a series of housekeeping genes or reference genes that we used. These are stably expressed in every cell of the body, so that the level is always constant all the time. So they're usually a good gene you can use to normalize your expression data, and I'll show you what that means in a minute. But then we looked at gene expression using a quantitative real-time PCR technique. I'm not sure if you've learned much about that yet, but I'll talk briefly about that technique. So, and we basically use that to look at the gene expression. And so very briefly, a quantitative real-time PCR is like a normal PCR reaction, which is PCR is really polymerase chain reaction to amplify DNA, particular stretches of DNA of interest. And quantitative real-time PCR uses fluorescent tags that enables you to quantify how much DNA is being produced each cycle in a way. And so it's a normal PCR machine with a fluorescent detector on the top. And as the amount of DNA increases, the fluorescent detector can pick that up. And so if you're doing an experiment like this, you've got a 96 well plate that looks like this. You've got all of your embryos here of different sizes and in vitro and in vivo produced embryos. And then you've got these two sort of parallel controls. These are 5-fold diluted serial dilutions of known amounts of DNA. And basically, you look at each of these one gene at a time. And when you put it into the machine, as the DNA increases in number, the fluorescence of those, oh, pardon me, the fluorescence of those standards increases. The most concentrated DNA standard increases first, and then a few cycles later, the second most concentrated, and so on. And so you've kind of got this beautiful standard curve that you can use to quantify the DNA amounts in your unknown embryos. So you might have an embryo that starts to amplify here and will produce a signal somewhere between these two standards here. So using that standard curve, you can calculate the amount of gene product that's being produced, essentially. All right, and so we did that for the embryos. We looked at a whole lot of different genes. And what I've just shown you here are those stable housekeeping genes that each of the embryos produces. And so this is the smallest embryo we collected, about 126 micrometers. This is the biggest one we collected. That's 680. That's an expanded blastocyst. This is a morula. And not surprisingly, when you look at the gene expression here, essentially, as the embryos get bigger, they express more genes, or the gene expression goes up. That's normal. That's not surprising because the embryos have more cells. So every single cell cumulatively is giving rise to these increases here. Okay, so this is going to be a bit of a problem if we want to actually look at gene expression changing within cells. Okay, and that's what you expect as the embryo develops. Certain genes will turn on and certain genes will turn off. But if the embryo is getting bigger, you won't see those differences unless you control for the size of the embryo. And so you need to kind of normalize your data to compensate for cell number. And so for us to do that, we used normalization against these housekeeping genes. So basically, to sort of, I'll come down here and sort of show you. We basically took 3 of those 4 housekeeping genes, and we used these to help normalize the raw data of all of our genes of interest. Okay, and so if you take those 3 genes, you take an average of them, and then you take the value from a gene of interest, divide it by these 3 reference genes. What it essentially does is it removes the effect of size. So here are all those embryos again, arranged from the smallest one over here to the biggest one over here. And this is for a particular gene, SRP14. All of the data has been normalized against those 3 housekeeping genes. And now that you see there's no size effect, all of them produce exactly the same amount of gene expression for that housekeeping gene SRP14. Okay, same thing for RPL4, another housekeeping gene, PGK1. Also, they're all really constant production. So our normalization is really good. When we get to this gene here, it's a little bit wobbly. And then when we get to CDX2, that one is really wobbly. And we know this one is actually a developmentally regulated gene. So it should change with development. Okay. So just to follow this through, these are all in order individual embryos. If we group them by developmental stage, so here's our morula, early blastocyst, expanded blastocyst, for the same gene, there's no significant change during development for this one, or this one. Here's that housekeeping gene that should be stable, but it's actually decreasing as the embryo develops. So that housekeeping gene is actually not a very stable gene to look at in this study. So we excluded it as a gene. And this CDX2, that's our trophectoderm or trophoblast gene, that's actually increasing with development. So essentially, that's what we expect. We expect gene expression to go up there. So this kind of helped confirm that our normalization was doing the right things. All right. There's just a bit of nuts and bolts behind that. Don't get too worried about all this. Let's now, using that background, let's now have a look at the results that we got when we look at those 4 genes of interest, really. The genes for the inner cell mass, the genes for the epiblast, these are the genes for the outer trophectoderm, and of course, this is for the hyperblast layer that forms the future yolk sac, okay? And there's a lot on this slide, so I'll walk you through those particular graphs, but the general pattern of all of these genes suggests that it fits with the role of the formation of the inner cell mass and the trophectoderm and the subsequent formation of the epiblast and the primitive endoderm. And I'll sort of show you that now. But if we go here and we look at OCT4 and we look at NANOG here, both of these are significantly downregulated between the early blastocyst stage and the expanded blastocyst stage. So they drop for OCT4 and they also drop for NANOG here. And that's around the time that the inner cell mass is actually forming, or it's forming, and then it's turning into the epiblast. So once that's happened, the genes turn off. Whereas CDX2 here keeps on increasing. And that makes sense because there's an ever-increasing number of those trophoblast cells that are getting bigger and bigger, while the inner cell mass is getting smaller little clump of inner cells. If we look at what have we got here, GATA6 now, GATA6 basically is expressed early and turns off as soon as the cells differentiate into hyperblasts. So it's not required to stay on for that primitive endoderm or that hyperblast to persist. Okay, so this is kind of the normal pattern. Now it's going to get a little bit more complicated. Here's our in vitro produced embryos now, and I've got them on the same graph to compare to the naturally conceived ones. But again, I'll walk you through this so that you understand it. So let's look. But first, just as a summary, the first thing that to take away from this is that the pattern of expression of the in vitro produced embryos is altered. It's not normal. And what it shows is there's a delay in the development of these embryos. So they're not forming the inner cell mass, the trophectoderm or the primitive endoderm in a timely way. They're actually delayed. And so just to look at this, if you look at NANOG, for example, normal expression is downregulation from this early to expanded blastocyst stage. NANOG in the in vitro produced embryos actually starts to downregulate early in some ways. That's why there's a significant difference between these two groups, which are the early blastocyst stage embryos. So some things are happening a little earlier. Things like GATA6, for example, which should start off really high, in vitro produced embryos, only 2 out of the 5 embryos even produce the gene. So not all of the embryos are producing the gene, and there's no change during development, whereas it should be significantly downregulated. So there's no change in expression that's happening. And then on top of that, CDX2, we couldn't even measure it in these in vitro produced morula stage embryos. It was not detectable. And the other thing that made the whole thing quite complicated was the normal pattern that we saw in the naturally conceived embryos where OCT4 goes down and CDX2 goes up. We actually don't see that in vitro embryos. All we see is no change at all in OCT4 during development. And then when CDX2 does come on for the first time, it doesn't keep on going up. It just stays the same. So all of this stuff is pointing to the fact that gene expression is not changing much in vitro produced embryos, which means they're not differentiating when they should. And so I guess to sort of summarize this, firstly, if we just look at looking at our in vivo embryos and what have we learned about the horse compared to the mouse. This diagram here kind of shows you what to expect for the horse gene expression. And it's very similar to the mouse in that it looks like OCT4 is coming on in the inner cell mass. It's driving the development of that inner cell mass, whereas CDX2 is coming on and driving this differentiation to the trophectoderm. Subsequent expression of NANOG here, that those cells will go on and form the epiblast, which is this red layer. And then these green cells here that are producing GATA6, those very briefly will turn on, form the primitive endoderm or the hyperblast, and then turn it off. So that seems to be kind of close to the mouse. The second finding about the abnormal gene expression in the in vitro produced embryos says that those embryos look like they're developmentally retarded. So they're not developing normally. And so they're delayed in their formation of the inner cell mass in the trophectoderm, that chorionic layer, the placental layer, and also the hyperblast layer, which is going to form the yolk sac. And so that's a bit of a worry, because one of the unique features of the horse embryo, and I mentioned this a little bit earlier, is around day 12, these embryos have to move rapidly around the uterus, about 14 times a day. They cycle across the uterus and into the different uterine horns of the horse embryo, of the horse uterus, pardon me, and they secrete those maternal recognition of pregnancy signals to the mother. And what that does is it keeps the corpus luteum on the ovary alive, and it keeps progesterone going to maintain pregnancy. So the problem here is if you have a delay in embryo development and the trophectoderm is actually delayed in its formation, it's not able to produce those maternal recognition of pregnancy signals in time, in this case being estrogen. The other thing is if these embryos are delayed, it's possible, although we don't know this, their ability to migrate around the uterus is affected as well. And so that means estrogen is not being secreted locally and not stopping the formation of a substance called prostaglandin F2 alpha, which will act on the corpus luteum and break it down. So what that means is that delayed embryos are at an increased risk of being aborted in the pregnancy. So the pregnancy could be lost because the embryos are not developing normally, okay? So that's a real concern. And in a way, it sort of explains why in vitro produced embryos only have 50% pregnancy success compared to 20%, compared to 80% naturally conceived embryos. So it's a bit of a problem. Now, another interesting observation, we're almost getting to the end of the talk now, but is that if you take those in vitro produced embryos, even though they're developmentally delayed, you can actually produce offspring, by 83% of foals will be born if you take those day 7 in vitro produced embryos and you actually transfer them to recipient mares, so mares that are ready to receive an embryo, but they're day 5. So they're actually 2 days earlier than a day 7 embryo. And so you're transferring a more advanced embryo to a less advanced recipient. And what that does is it gives the embryo 2 extra days in the uterus before it has to, it gives it 2 days to basically develop further and catch up and develop normally. And so what we're finding is if you do that asynchronous embryo transfer, so an older embryo to a younger mare, you can increase your pregnancy rates and your offspring production rates. And what we think is going on here is that the in utero environments is actually giving those in vitro produced embryos a chance to sort of catch up and develop normally because they're not developing well in the petri dish. The uterus is much better, more natural. And just to sort of prove these effects here, a couple of studies have looked at the expression of that OCT4 gene in the embryos, looking at the protein expression. And so this early study shows OCT4 in the inner cell mass down here, but it's also weakly expressed in the outer trophectoderm as well. But that expression will go down once the embryo gets to about 600 micrometers in diameter. Now, another group in the US did a great study where they took in vitro produced embryos and they looked at the expression of OCT4 in red here. So you can see all of the nucleated cells here brightly expressing OCT4, okay? This is an in vitro produced embryo at day 11. So it's been in IVF for a long time. I'll get to your question in a minute. And so in the normal situation, if you don't transfer these embryos to the uterus, the inner cell mass here shows OCT4, and all of this other tissue out here is the trophoblast. That's also showing OCT4. If, however, you produce an in vitro produced embryo, stop it at day 7 and transfer that to a recipient mare for 3 days inside the uterus, and then recover the embryo again. So this is now a day 10 in vitro produced embryo transfer embryo that's been in the uterus. Here's the staining for the nuclear cells. When you look at OCT4 expression, it's only confined to this side here where the inner cell mass is, and all of the trophoblast cells, pardon me, shown by the nucleus here, are not expressing OCT4 anymore. So all of a sudden that transfer to the uterus can actually help kind of normalize these embryos and improve their chances of foaling, of pregnancy success and pregnancy to term. So that's sort of the end of where we've finished so far. But for us, in terms of where we're going from here, really, if the uterus can actually improve or cause improved survival of those embryos, perhaps we can actually look at the genes that are being expressed and do some more real-time PCR expression of those embryos to see if the gene expression pattern actually becomes more normal, for example, if they actually show evidence of catching up. And I think the other thing that we've sort of demonstrated is that these four genes are quite sensitive and they can be used to detect all sorts of abnormal environmental effects on those horse embryos. So we've shown that it works for in vitro production, but animals that are experience heat stress or animals that are older, so older females, the embryo quality can decline because the oocyte quality declines. Maybe we can use these genes to detect if there are abnormal differences there as well. And I guess because to some degree, horse reproduction and human reproduction is similar, particularly in terms of these maternal age effects and what that has on the viability of embryos, it may well be that a horse could actually be a good biomedical model for human infertility studies as well. So I'll leave it at that. And I just want to thank my colleagues at both the University of Utrecht, the Hubrecht Institute, and the group in Italy that helped produce those B.SC embryos for all of their help in doing this study. So that's it. And thanks for your attention. Happy to take any questions if you have them.
All right. Thank you so much for your wonderful presentation. Yeah, I got a lot of knowledge from you. And if we look at the chat box, then there are a couple of questions from the student and also I think also from the lecturer here.
Uh-huh.
So
Quick look.
let's check it out one by one.
Okay.
Uh,
Happy to
there's
work
one
through some of
question
those.
from Tirza Veronica, I think. She's asking like how long is the process of its steps from morula, blastula, gastrula in horse. So
In
that
horse, yeah.
yeah, so if we are working on IVF in horse though, so that we know how long we can manage the time for morula, blastula, gastrula,
Yeah,
and
yeah, that's
also.
a very good question. I'll pull up that slide again because there's actually, if I can do that quickly, and I'll go backwards because if it's going to get me there, hello, 1, 2, 3, because I showed a little bit of that in one of the diagrams at the start, just we're almost there. And 1, 2, this one here. So essentially, your morula will start to form at about day 6. Okay. Then it rapidly develops to an early anablastocyst stage. So we had a lot of difficulty recovering morula stage embryos because they just enter the uterus, and then within half a day they already differentiate into an early blastocyst. So we got 5 morula, but we must have flushed 15 mares to get 5 morula because every time we flushed them, we checked it on day 6 and we were like, we're too late. It's already an early blastocyst. So this, for those stages of development in the horse, you've really got basically 1.5 day difference in the development. And then the gastrulation is really occurring out here at day 12. So you can culture embryos in vitro to these stages, but once they get to these stages, they get quite big and they're more difficult and more delicate to manage in vitro. And I think the problem also with the in vitro embryos is if that capsule doesn't form properly, then they'll probably rupture. So in a way, your in vitro window of development before you want to transfer those embryos, you're pretty much stuck between day 6 and probably day 8, day 9, and then you wouldn't want to keep something much longer because if you wanted to transfer it again afterwards, as they get bigger, the chances of them rupturing are higher. Good question.
All
I'll
right,
stop again.
thank you. Also, another question from Otnil Budi Kristianto. Do you want to ask yourself? Mau nanya sendiri Kak ke Prof. Damien?
Sorry, say that again.
I'm talking in bahasa Indonesia because
Oh, yeah.
someone is asking in bahasa Inggris, in English. So
Okay.
probably he want to ask to you directly.
Please go for it.
Iya. Otnil Budi Kristianto mau nanya sendiri nggak? Oh, no. All right. So I will read the question.
Okay.
He asks you that I would like to ask about the in vitro production method. Why
Right.
is the conventional IVF method considered ineffective in horses?
Yeah.
And why does it require the use of the ICSI technique instead?
Yeah. That's a great question.
Mm-hmm.
Um, it's unfortunately I haven't worked in that space myself, but I know from the literature that they've probably the equine field has been trying to get conventional IVF to work for probably about 1 decade, so about 10 years. Um, and of all of the studies that have been done from some of the top equine labs, they've only been able to produce 2 offspring by IVF. And one of the reasons it seems to be a problem is the in vitro environment in the horse is still not optimized. And so when you put sperm in the fertilization dish, pardon me, um, there's difficulties in capacitating the sperm. So because it's not in the female reproductive tract, it's got to go through those developmental changes so it can fertilize the egg. And it's difficult to do that in vitro with horse sperm. And the other problem that seems to happen with the oocytes is when you put those in culture, in a very short period of time, the zona pellucida on the outside actually starts to harden. So even if you capacitated the sperm, they're not able to digest through the zona anymore. So in a way, some sort of progress or breakthrough needs to happen to improve the culture conditions for the horse embryo, so that natural IVF can happen. Or I say natural IVF, but it's natural fertilization at least. So because of those problems, if you just take the sperm and inject it directly into the egg, you don't have to worry about capacitation of the sperm, and you don't have to worry about the hardening of the zona. So it's just become a more practical approach to get around those problems, unfortunately.
Yeah. So
Thank you.
yeah, so it's because of limitation in the sperm function in that horse, right?
One is yes, like the ability to capacitate the sperm. And the second is the zona pellucida around the oocyte seems to harden unnaturally, almost like the polyspermy block. It tends to harden so that the sperm can't penetrate. And that's not because it's been fertilized, it's because the something in the culture conditions, the constituents cause the zona to go hard. So that's means that there's a permanent barrier around the oocyte as well.
All right. Thank you. Yeah. Do you have more question, Othniel Budi Kristianto? It seems clear for you? All right. It seems clear. Right. We move to another question from
Mhm.
Pak Andi Saputra. He's asking, how does the process of in vitro fertilization influence the early embryonic development in horses compared to natural fertilization?
Yeah. So, I mean, at least what we found from our studies and what others have shown as well, I guess from some of those fluorescent images I showed you of the embryos, it looks like the in vitro produced embryos, they're just developmentally delayed. It just takes them longer to go through the 2-cell, 4-cell, morula stage, blastocyst stage. And so they're delayed, which is a bit of a problem because implantation and maternal recognition of pregnancy signals is very precise in its timing. And so if you have embryos that are in a uterus that are delayed, they're not going to produce the signals at the right time, and then the pregnancy will be lost. So that's one complication with these embryos. Um, uh, the other thing that I showed you, I think, in the slides was that some of these important structures that need to form, especially because the embryo has to move around the uterus, like the capsule, the glycoprotein capsule, isn't forming properly. And so what that means is that those embryos, the in vitro embryos, can be in the uterus, they can try to move around the uterus, and if they're not protected by the capsule, the cells can break apart and the embryo will end up degenerating. And that also might contribute to why 50% of in vitro embryos don't, they might start a pregnancy, but they don't actually go to term. So there's definitely developmental delays and some abnormalities in those in vitro produced embryos. And it probably just means that the culture conditions are not optimal. We still haven't quite worked out the best conditions for those embryos.
All right. Um, this is my own question. Is it
Mhm.
happen to all horses or just the horse that you were working on?
Yeah, that's so. So is it that particular breeds
Mm.
that are different? That's a good question, and it's hard to say because I mean we've done these studies on they were Dutch warm bloods. Um, I think the only other groups that are working in this space, I'm trying to think about what breeds they were working on. Um, I don't know what breeds they were. Certainly, it's confined to like the studies we've done have been in Dutch warm bloods. There's been some breeds from Italy and some US. Those are the main groups, the leading research groups in this area. And so there's either thoroughbreds or quarter horses in the US. In Europe, in Holland, it's Dutch warm bloods. In Italy, I'm not sure what breeds they've got down there. So we're seeing that in those breeds. I'm not aware of any literature that shows that there are other breeds that are doing things particularly different. Um, it's just and it's not surprising. I mean, I think the in vitro environment, we saw the same thing in cattle, maybe 40 years ago when we were developing IVF in cattle. It wasn't very optimal back then, and so embryo transfer rates, pregnancy rates were quite poor. But there's been 50 years of research in cattle IVF and lots of embryo transfers. Now it's almost routine. So I think it's more a question of with increasing research, the efficiency will improve. And I think at least one of the I mean, some of this information is a little old now, so it's not the latest results, but some of the workarounds, including taking the day 7 embryo and transferring that to a day 5 recipient mare, if that gives you 83% foal rates, you've solved one of your problems in a way. So you can spend 10 years improving the media or just do a transfer to a more immature recipient animal and you'll still get the same result, I suppose.
All right.
Good
Interesting.
question.
Very interesting. Well, we move on to the next question from Linda from UNS. She's asking, how do we difference between the in vivo and in vitro environment that influence the early cell differentiation in mammalian embryo?
Yeah. Oh, those
Yeah,
these are
because
very good questions. Wow. Very good questions.
from bachelor, yeah, it's good question.
Yeah. Oh, gee, how do you do that? Look, it's a great question. I'm not even sure how to tackle that, but for example, if there are ways you can detect the in vitro uterine environment, if you can measure pH, for example, if you can measure the ions, sodium, potassium, what ions are present at the time, what growth factors might be present. And so some of those answers can come from maybe uterine biopsies of animals during different stages of their pregnancy. Early pre-implantation biopsies of the uterus might tell us something about the genes and the proteins that are being produced at a certain time, and that can help us mimic those conditions better in vitro. If we know there's a certain growth factor that the uterus produces at a certain time, then we should make sure that that growth factor is in the IVF media, and hopefully that will get us over some of those developmental difficulties that we're seeing. And certainly I know that has been the approach in solving IVF problems in the cow, solving IVF problems in mice originally. We embryos would develop to 2 cells and then they would stop, and we called it the 2 cell block. And then they did some research and they realized, oh, we need these growth factors. They added the growth factors, the embryos went from 2 cells to 8 cells, and then it stopped again. And then they had to work out what was the next bit. So I think because the horse, it's not as commercially valuable as the cow, for example. There's less research and less teams working in that space, so it will still take more time to get there. But those would be the approaches I would think you might use to actually improve the culture conditions.
Mm.
Great question.
Yeah, 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 yeah. It seems like the students are really excited with you. They have so many questions.
Oh, fantastic.
Okay, a question from Amira. She's asking, is the ICSI method effective in increasing the success rate of artificial insemination to save endangered species like rhinos or horses or any animals?
Yeah, yeah, that's a great question, and that this is probably an area that I have probably more recent experience with than the horse stuff. Look, I think with any of these assisted breeding technologies, um, you need a lot of you need to learn a lot about the species that you're working with, and so the more invasive the technique, the more knowledge you need to have about the oocyte, the sperm. So if I think about it this way, if I think about artificial insemination, what you need to know is how to get the sperm from the male, how to keep it in media to keep it alive, and then how to, if you need to, well, it should, if you're inseminating, you don't even need to know about how to capacitate the sperm. You just need to collect it, keep it alive for a little while, transfer it. And in the future, you want to improve, you want to develop a freezing technique so you can freeze it down into a sperm bank. That's a lot of work there already, but it's not too complicated. With IVF, you need to know all the sperm sides, but you then also need to know how to collect the egg from the female. You need a surgical technique to get the oocyte. You need to develop a culture condition so that you can mature the oocyte so it's ready to be fertilized. You need all of the techniques you need for sperm. You need the right culture condition for fertilization. Then you need the right culture condition for embryo development in vitro, and then you need a surgical technique to get it back into the animal. It's 10 times more complicated. So in a way, if you're working with a species where there's nothing known, then your best chance is using the most low technology approach because it requires the least knowledge and probably has the best chance of success. So for us, for example, we're working with African wild dogs, and the most logical approach is just collect sperm, freeze it, thaw it, inseminate into the female. And all we need to know is the time of ovulation, and we could just inseminate the sperm into the uterus, and hopefully, we get pregnancies. If we did IVF, it would be more complicated. So, I think that's the easiest approach. But there are other experts working in the rhino field, for example, at the moment, that are trying to develop IVF. And so they're actually developing techniques to collect eggs and then fertilize those and transfer. It's very challenging. They're doing great work, but there are not many people that can do that work. It's so specialist that it's hard to make a broad impact around the world if only two people in the world can ever do that experiment, that procedure. So low tech in my mind, is always better to have an impact in the wild, I suppose. But yeah, a good question.
Oh, right. Thank you. There's one question from the lecturer,
Okay.
Devi Puspitasari. She's asking, with your expertise, is it possible for you to do a wildlife conservation for the animal in Indonesia?
I mean, that would be lovely. I mean, I'm always motivated to do. I mean, I'm motivated by conservation of wildlife and using reproductive tools to help wildlife. That's my passion. That's what I did my PhD in. And so, if I think if there's a species in Indonesia that is a good candidate for these approaches, then that certainly I would be interested in trying to make a contribution. It should be led by you guys, but I would be happy to actually help if that was possible. Um, so yeah, so I mean, it comes down to recognizing what species you could make a difference to. And again, my suggestion would be low tech first. So keep it simple.
Yeah.
Freeze sperm, artificial inseminate. You can move genetic diversity from one population to another. That sort of stuff has the greatest chance of success. When we start talking about IVF, or we start talking about cloning and all this other stuff, and there are lots of groups doing cloning at the moment, but it's from a conservation point of view, it's a little bit science fiction. It's not going to save the species, and that's what we should be focusing on. So, yeah, if you identify species of value, then we can discuss what might be possible.
Thank you. One last question, I think, from Pascalia Apriani. Oh, no, it's not last. Well, she's asking,
Mhm.
do in vitro produce horse embryo? Oh, wait.
Okay.
Do in vitro produce horse embryos show any adaptive mechanism to compensate for the artificial culture environment?
Yeah. It's again, that's a good question. I mean, all I can say is from the information that we've seen with these embryos, we know they're delayed, so that's a problem. But it looks like they're not permanently delayed. You can actually get them back to normal if you put them in an improved environment. So that stuff I showed you at the very end, where we spoke about transferring the embryos back into the uterus and letting those in vitro produced embryos spend time in the uterus, it seems to accelerate their development. So if their delay is there, it's not a permanent problem. It seems to be they seem to just grow poorly in vitro. So if you can generate the embryos and transfer them relatively quickly, or transfer them asynchronously to a more a less advanced recipient animal, then the embryos have enough time to kind of catch up. So they're not permanently damaged, it would seem. You just have to give them more time, or get them out of the in vitro environment quickly, or improve the in vitro environment. One way or the other, they're able to recover, it seems.
Okay. Another question. On your slide,
Hmm.
you mentioned that after ICSI, you need to add chemical. So what
Oh,
chemical is it?
yeah,
Uh,
yeah,
that you use it to activate the oocyte.
yeah. Very good. Yeah,
And is it always produce a good quality embryo because of that chemical? Because
yeah.
in human IVF chemical activation, she using calcium ionophore, which is
Yeah.
sometimes produce poor quality embryo. So what chemical that you are using?
Yeah, so that's a great question, and that's it's your student has sort of answered the question. So calcium ionophore is normally used to cause that surge of calcium uptake in the oocyte, which mimics normal fertilization. And you only have to do it because if you're using ICSI and you penetrate the oocyte hasn't gone through that natural fertilization process itself, which would normally cause an influx of calcium and cause the normal development program to begin. So yes, it's the same as what's being used in the human space, a calcium ionophore to actually cause an influx of calcium to start the activation process. I'm not sure whether or not it's I'm not an IVF expert, so that's the thing. I've done the gene expression work, but I haven't done all of the culture work. That was done in Italy. But I'm not aware. There might well be alternative approaches that you could use to activate oocytes after fertilization by ICSI. And I don't know whether or not there's electrical stimuli that you could provide, which would do an alternative to calcium ionophore treatment. Um, but yeah, that's the approach. That was the approach that was used in the horse. So calcium ionophore to cause the activation. I don't know um, if I don't have the information on what effect that has in terms of the quality of the embryos that come from that, because they're all activated the same way.
All right. Atia Nurfadila, if you want to talk to Prof. Damien directly, you can just raise your hand and say something. Atia Nurfadila, silahkan kalau mau nanya. No? Okay, then I will read it. Anyway, another question is, the students are really excited with you.
Okay. Okay.
She's asking, in the context of equine embryo development stages, Does the impaired sperm function in horse, which leads to IVF failure,
Mm.
also affect the early embryonic mRNA transcripts that determine the successful formation of the epiblast and hypoblast?
Yeah,
You
so
get the
so that,
question?
I think I understand that question. I'll just repeat it to be sure. But so I guess it sounds like the question is asking if you have subfertile sperm,
Mm-mm.
does that translate through to poor mRNA expression in the developing embryo because it's basically being inherited from the sperm in a
Yeah.
way, I suppose. Yeah, that's a great question. I don't know in the horse at this stage, so I can't say to you. I mean, normally in sperm, so there's virtually no mRNA transcripts in the sperm itself, so it all comes down to the transfer of the DNA package itself. But when the DNA is transcribed, obviously, if there are any defects in the DNA, that will affect the mRNA that's produced from the paternal genes. Okay, because usually the oocyte contains most of the messenger RNA, all of the proteins and all the goodies, and the sperm predominantly contributes DNA. Okay, not so much transcripts and so on. But I can't answer this in the horse, but we have done some work in pigs where one of the things that we showed was if you have sperm that has high levels of DNA damage, so broken or fragmented DNA, even though the sperm looks normal and it swims normally, looks like every other sperm, if it fertilizes the egg, when the embryonic genome turns on, so both the sperm genome and the oocyte genome turn on, then gene expression is abnormal and the embryos don't develop. So there is certainly an effect, not necessarily at mRNA initially, it's transmitted through the DNA itself. If there's damage in the DNA by fragmentation, that will affect the embryos. So sperm quality in the male is actually important in uh, uh, contributing to the embryo's success.
All right. Okay, another question from Eka. She's asking, in the equine embryo development, what is the factor that will significantly change the morula in the day 6 to plastocista in the day 7?
Yeah. So we at least we know from the work I've done. I haven't seen if I'm not sure if there's more stuff that's been published since, but from the work that we've done, it looks like those genes that are involved in the mouse also seem to be involved in the horse. So OCT4 seems to be the gene that drives the development of the inner cell mass. CDX2 is present. It seems to be the gene that controls development of the outer trophoblast. Nanog is present and it turns on about the right time. It seems to be involved in the formation of the epiblast, and the GATA6 is expressed at the right time and seems to be involved in the formation of the hyperblast. The work would be advanced further if we could actually also do some protein staining studies where we could actually see that it's coming on in the right cells at the right time. At the moment, we know the genes are going up and going down, but that's just across the whole embryo. If we can see where it comes on in the specific cells, then we can tick a box and say, yep, perfect. It's in the inner cell mass, only in the inner cell mass. That's the right gene. Excuse me. Apologies. Yeah.
All right. Thank you, Prof.
Very good questions.
Yeah. Right. So if no one wanna ask more question, so probably we can finish it. So before we finish, would you mind please to open your camera so that we can make a photo session together? Guys, please open your cam. Right, thank you. Oh, Budelia. She's my boss. She's the head of biology department. Ibu Yeti Hastiana. Makasih sudah bergabung. Thank you for joining. Right, so we are going to make a photo session. So I will let you know that there are at least 80 students joining
Oh,
this
wow.
afternoon.
Okay.
I mean, this is weekend, but all the students are excited to join your class, Damien.
Oh, thank you very much. Thank you. I'm honored that many people showed up. Thank you so much.
Yeah, okay, I'll take a photo for the first slide. There are four slides that I'm going to take a photo. So
Okay.
the first is the first slide. So please open your cam and I will take the best photo of you. I'll count it. One, two, three. Thank you. I'll put it on the slide. Be prepared for the second slide. Second slide. One, two, three. Thank you. I will take another photo for the slide three and four. There you are. Third, third slide. One, two, three. Right. And I will take another photo for this last slide. Slide four. One, two, three.
Can we do one more photograph where we actually wave or do something like that?
Of course. Like what? Okay, like this.
Yeah, like this. Okay, that's
If
good.
you
That's good.
like it, then I will take a photo. So please, guys, gini ya. What is it? Is it IVF or something?
Oh, I don't know. I don't know. Just hello, everybody. That's
They're like,
fine.
hi. Okay, I'll take another photo of you. One, two, three. Yeah, so cute.
You. Thank you. Oh, I'm glad I'm shaved this morning because I now I know I'm going to be on camera. I'm glad I cleaned myself up.
All right. So thank you so much, Prof. Damien. But it's so that's really I got so much insight
Thank
and
you.
knowledge also for the student as well. And the lecturers are also joining. So what a wonderful weekend. So productive.
Thank you. Thank you. And thanks for the attendance and for getting to meet you all. And good luck, students, with your studies.
Yeah.
And hopefully I'll see you in the future.
Yeah, thank you. So everyone may leave. Thank you. Thank you, Damien, for
Okay.
this.
Bye. Thank you.
Bye.
Thanks, Anita. Bye. Bye.
Ya,
Bye, everybody.
yang sudah ngisi absen boleh leave. Yang ketinggalan absennya, saya tulis di chat. Ketika teman-teman ngisi absen, itu nanti dapat sertifikat sebagai kenang-kenangan kebersamaan kita di weekend ini. Terima kasih sekali lagi. Sampai jumpa pada guest lecture berikutnya. Wassalamualaikum warahmatullahi wabarakatuh. Presentasinya itu PPT nggak dikasih sama Prof. Damien. Jadi mohon maaf Ibu Yeti. Tapi saya ngefoto semuanya. Setiap slide saya foto. Nanti saya gabungkan dan akan saya kirimkan ke Bu Yeti melalui Bu Dewi.
Izin ya Mbak, tadi saya telat masuknya.
No, it's okay,
Udah
Pak.
kuliah dulu.
Oh, terima kasih.
Materinya bagus sih karena dia mengaitkan relevansinya dengan konservasi. Kalau bidang
Nah,
ibu kan, bidang saya kan lebih kepada environment. Jadi bagus itu. Saya nggak bisa ikut dari awal. Udah di separuh, nggak apa-apa ya.
nggak apa-apa. Ada recordnya Ibu, insya Allah nanti akan kami
Begitu.
share. Ya.
Tolong di-share ya Mbak Nita.